Okay, this presentation is about chemolithotrophy. So what is chemolithotrophy? It refers to a type of metabolism where cells acquire energy through oxidation of inorganic compounds. Certain organisms get their energy from inorganic compounds like sulfur and ammonia, and these compounds can also use carbon dioxide as their carbon source. The energy yield is lower than that of glucose molecules compared to if they were to use organic compounds for metabolism.
The process can use oxidative phosphorylation, but what is oxidized is the inorganic compound, which acts as an electron donor as well. The electrons go through the ETC, the electron transport chain, and generate a proton motor force that is used to generate ATP. Chemolithotrophy involves the generation of ATP and the generation of NADH, and the electron donors are oxidized in the cell, and the electrons are channeled. Electron acceptor can be organic or inorganic, and the hydrogen compounds used in the process are high in energy.
So here's just an overview of the process of how they get ATP through the inorganic source and a carbon source. So chemolithotropes can be aerobic, which means they use oxygen, or they can be anaerobic. Aerobic is preferred because it would generate the highest free energy yield and will generate the most ATP. So for inorganic substance, they always start with its inorganic choice of compound, and the electrons are transferred, which will generate ATP, giving a different form of that same substance. And then you will get your, depending on what type of anaerobic organism it uses, the aerobic or anaerobic.
There are 4 types of electron donors. These are the first is hydrogen oxidizers. These use hydrogenase enzymes to oxidize hydrogen gas. There are sulfur oxidizers. These oxidize, reduce, and partially reduce sulfur compounds. These would include hydrogen sulfide, elemental sulfur, thiosulfate, sulfite, and sulfate. Nitrogen oxidizers usually starts with ammonia, nitrification, and it's a 2-step process where the ammonia is oxidized to nitrate, NO2, and then it is further oxidized to nitrite. And that's the process of nitrification. Iron oxidizers oxidize ferrous iron to ferric iron, which goes from a 2+ to a 3+. And since ferrous is not a favorable reduction, that would make it very difficult to do.
So these are some examples of organisms. At the top, this is the allicogens. They convert H2 to water. These are gram-negative aerobic bacteria. Over here, this is the nitrosomas. They convert nitrate to nitrate. They're also gram-negative ammonia oxidizing bacteria. Right here you have the Theobacillus. They're also gram-negative. And here are the Begatoa. These are actually some pretty big bacteria.
Examples of organisms, these are the electron acceptors, and that would be this bacteria would be the methanogic bacteria, like the Methanostarcina barkeri. This would be carbon dioxide to methane, and also oxygen to water, which is in a wide range of different organisms.
So two examples of two processes. One is hydrogen oxidation. In bacteria, hydrogen can be oxidized by hydrogenase, which is just an enzyme that catalyzes the reduction of hydrogen to protons. This process causes proton pumping via electron transfer to various pronones and cytochromes. So they have this thing called canalogous bacteria, which are bacteria which can oxidize hydrogen. These bacteria can live in extreme conditions and are a mixture of both gram-positive, gram-negative bacteria. Some examples are the Hydrogenobacter thermophilus, which are gram-negative rod-shaped bacteria, and the Helicobacter pylori, or also gram-negative bacteria that is found in the stomach. It's very abundant in the stomach. Hydrogen can be used as the energy source aerobically or anaerobically. And in some organisms, they will use a second hydrogenase that is used to generate reducing power for NADH, which is used to fix the carbon dioxide.
An example of sulfur oxidation. Sulfur oxidizing organisms use a Calvin cycle to reduce carbon dioxide. It uses a reverse electron flow, which pushes electrons against their gradient to produce NADH. Inorganic sulfur is stored until needed. Sulfuric acid is a better electron donor, which allows more protons to be translocated across the membrane. It uses sulfur oxidation for energy and CO2 as a source of carbon. Organisms use sulfur oxygenase and sulfite oxidase to oxidize sulfur compounds. Begiatoa, a sulfur, and the picture on the right would be Begiatoa. It is a sulfur oxidizing bacteria found in marine or freshwater environments.
So this is just an overview of how sulfur gets reduced by chemolithotropes. So yeah, and how they generate ATP and NADH using that reverse flow for the NADH, and the CO2 with ATP for the cell material, and it just shows all the different transporters in the membrane.
Chemolithotropes are found everywhere in the environment and play a big role in many processes. Nitrosomonas are one example that is crucial in maintaining soil fertility in agriculture.
And that is the end of the presentation.
I hope you learned a little bit about chemolithotrophy.